Welcome to Chapter 37, Plant Sensory Systems, Signals, and Responses. By the end of this chapter, you should be able to describe how plants detect sensory information, explain the connection between signal transduction and hormone production, summarize how hormones regulate plant growth and responses to the environment, and explain how plants defend against pathogens and herbivores. So if you're a plant, you're just hanging out there, anything like a wavelength of light, or touch, or some kind of a binding molecule can serve as an environmental stimulation. Plant sensory cells have signal receptors that can change shape in response to these stimuli. So in this example here, we have a leaf, and it's getting a little bit of some kind of a stimulus on the leaf, right? So it is touching the outside of the cell, and then that's being converted into an internal signal. So the signal is being transduced from an external signal to an internal signal, and then a cell-cell signal, as that cell produces hormones to transmit the information to the other cells. And then that kind of a cell-cell signaling hormone is released eventually in the cell that needs to respond to the message. And that cell will do something, like grow in a particular direction, okay? So this is the process by which an external signal becomes an internal signal within the plant. So how does the information from an activated sensory cell actually get to its target cell? Hormones. So these are molecules that are produced somewhere that do their work somewhere else. A hormone is transported to the target cells where it causes a physiological response. All right, so the first response we're going to look at that plants do is the phototropic response. And actually, some of the earliest work on this comes from Darwin and his kid. So they kind of did a father and son project in which they germinated seeds in the dark and then placed the young straight coleoptils, so coleoptils are those growing shoots of the plant, next to a light source. And so the coleoptils grew towards the light, so that's phototropism, right? So it's just a plain coleoptil, the light's here, it grows in this direction. We know now that this happens because the cells that are on the shaded side elongate, but Darwin and his son did not yet know that. They also found that plants only bend towards light with blue wavelengths. So they tried to use lots of different kinds of colors of light. So they tried blue light, yellow light, green light, orange light, and red light, but the plant only bent in response to the blue wavelength light. So there are photoreceptors that detect the blue light and initiate this phototropic response to cause the plant to bend. And so those photoreceptors are called phototropins. So the Darwins carried out additional experiments to figure out the details of how all of this is working. So they took the top off of the coleoptil off of the plant. So this is the control. It bends towards the light. They cut off the tip, and when that happened, it didn't bend. They tried leaving the tip intact but covering it, and they also found it didn't bend. They left it uncovered but covered up the part that was doing the bending, and it still bent. Okay? So their hypothesis was that light is sensed at the tip of the coleoptil, and the null is that it wasn't. And after doing this experiment, they were able to interpret, okay, so if you have light coming at the coleoptil, the tip of the plant, the growing tip, it is somehow able to sense the light and send a hormonal signal farther down, and then cells over here responded by growing longer. So again, the Darwins proposed that there's a substance that's produced at the tip of the coleoptil that acts as a signal that is transported to the area doing the bending. So additional experiments done by Fritz Wendt worked on this. So having decided that there is indeed some signaling molecule that moves from the tip of the coleoptil to the cells that are doing the bending, Fritz tried a couple of things. So he tried to use a little block of agar. So he cut the tip off of the coleoptil and then stuck the agar on and put the tip on top of that. And it still bent. So whatever molecules it was releasing were able to pass through this porous agar and go where they needed to go. When this was replicated using a thin sheet of mica, which is a rock, no signaling molecules were sent and no bending occurred. So we know that this signal is some kind of a chemical that's going through the plant, right? It's not something airborne. The plant has to have direct contact where the tip is in contact with the rest of the plant. All right, so then you exposed the agar blocks to different kinds of, let's see, did I say that part? Oh yeah. So you did different things in which you cut the coleoptile tip off, and then here's the agar block, and then you put the coleoptile tip on the agar block, right? So the tip is exposed to sun, and then cut off, and allowed a little bit of time so whatever signaling molecules are in the tip can go into the agar, and then just the agar is moved onto the plant, and that causes the plant to bend, but you can actually cause the plant to bend in different directions by where you put the agar block. So if you put the agar block off of this side, it causes it to bend this way, and then this side, it causes it to bend this way. So what this suggests is that the cells on the elongated side, or the cells on the shaded side, are elongating in response to the hormone. Okay. So Fritz Wendt named this first plant hormone Auxin. So Auxin is what we call that hormone that causes plants to bend in response to light. But later on, we went and identified the chemical formula for that, and so we now call, well, Auxin has two names. It's called Auxin sometimes, but it's also called indole acetic acid, or IAA, which is its proper chemical name. All right, so what physiological responses occur as the plant grows towards a light source? A, the tip of the shoot detects the direction of light, and the cells elongate on the shaded area of the shoot. I like that one! Well, let's see what else there is. The base of the shoot detects the direction of light. No! The cell division occurs on the shaded side of the shoot. That part's okay. The entire shoot detects the direction of light. No. And cell division occurs on the shaded side of the shoot. That part's okay. The base of the shoot detects the direction of light. No! The cell elongation occurs on the illuminated side of the shoot. No! The tip of the shoot detects the direction of light. Yeah! And the cells shrivel up on the illuminated side of the shoot. No! All right, so it was A. All right, so plants are sensitive to wavelengths in the red and far red portion of the visible spectrum. And this is a series of experiments where we do funny tricks with red light and plants. So red light drives photosynthesis, which you know what that is, and promotes germination. Germination just refers to sprouting, so making seeds sprout. But far red light, so if you shift the wavelength into the far red zone, it indicates shade and it inhibits or stops germination. So red light and far red light have opposite effects on photosynthesis and germination. All right, through experiments, people have figured out that the last wavelength that a seed detects will determine whether or not germination will occur. All right, so let's say you just expose the plant to red light. They germinate. Okay, red light, but then far red light, don't germinate. Red light, far red light, germinate. Red light, far red light, far red light, don't germinate. Red light, far red light, far red light, germinate. Red light, far red light, far red light, far red light, don't germinate. And then all that ending in, so basically, any sequence that ends in red light, it germinates. Any sequence that ends in far red light, it doesn't germinate, okay? Let's see, so plants also will change their growth patterns in response to photoperiod. So photoperiodism is any response by an organism that is based on the photoperiod, meaning how long day is versus night. So obviously, in most parts of the world, days are long in the summer and short in the winter. Days are long in the summer and days are short in the winter. And plants use that as a cue to decide when to grow and when not to grow. So we know that there's three kinds of plants. There's long day plants that tend to bloom in the midsummer when the days are long. There are short day plants that bloom in the spring or the fall when the days are short. And there are day neutral plants that flower whenever and don't really worry about photoperiod. So short day plant. So if you have a situation with a long night and a short day, okay, the short day plant will make flowers, right? And the long day plant, see, this is a short day, not a long day, so the long day plant won't make flowers. All right, let's just say you have a short day and then in the middle of the night you have a bright flash. The plant will somehow detect this whole period as having been daytime, and the long day plant will bloom, and the short day plant won't. All right, but what happens if you do red light flash, then far red light flash? Well, the plant's like, oh, I thought it was a longer day, but no, I'm wrong, it's a shorter day. Okay, and so then the short day plant flowers. What if you have a short day, but then red, far red, and then red light in the middle of the night? Oh, the plants are like, oh, it is really a long day, and the long day plant will bloom, and the short day plant won't. What about red, far red, red, far red? Nope, doesn't perceive this time as being daytime, so the short day plant blooms and the long day plant does not bloom. Now we're moving on to the flowering hormone. And so this is a hormone that induces flowering. And so we know that if you expose even one leaf of a plant to the conditions necessary to induce flowering, the whole plant can flower. And so we know that the flowering hormone, well, we've named it, it's called phloragen, right? So you can tell through experiments that phloragen can induce flowering. So the question is, can a signal produced in one plant be used to induce flowering in another? So the hypothesis is that a signal from a grafted leaf can induce flowering, and the null is that it won't work. So basically what they did is they took a plant and exposed it to a short night photoperiod, and this is a short, so it's short night, long, whatever, short night, long day. This is a plant that grows during short night, long day. And they took the leaf and they stuck it on this other plant. This is a day neutral plant exposed to a long night photoperiod. And then once this leaf was transplanted over here, that induces a plant to flower. So it's the signal coming from the other plant that induces this plant to flower elsewhere. So a signal from a grafted leaf can induce flowering. All right, the next response is a gravitropic response. And gravitropism is movement or growth in response to gravity. And so we know that roots will respond to gravity by growing down and shoots grow up. If you cut the root cap, remember that little mass of cells off the end of the root tip, the root will not respond to gravity. And so we know that the gravity sensing occurs in these cells that have starch granules in them that are at the center of the root cap. And so the statolith hypothesis explains how plants are able to detect gravity. So you have these cells here called amyloplasts, right? And they store starch, and so they will sink to the bottom of the root cap cells by gravity. Okay, so the amyloplasts are pulled down, and when they get to the bottom, they activate these pressure receptors. So if the plant is turned over onto its side, then they'll all go this way, and then they'll activate these receptors, right? So the position of the amyloplasts will basically tell the sensory proteins which way is down, and that initiates the gravitropic response. All right, so people did an experiment in which they took statoliths in a root cap and replaced them with iron filings, okay, little bits of iron. They then held a magnet on the side of the root as the root grew. Let's just say we hold it off over here on the right.
Which right?
You know, the right.
I don't know which is the right.
It's over here on this side.
Okay. Point.
Over here. Okay. What results would support the statolith hypothesis of gravitropism using this experiment?
I guess that the root should grow towards the magnet.
Yeah, the root grows towards the magnet. That is true. The root should grow towards the magnet, right? So the iron filings will fall against these receptors, and that will tell the root to grow in the direction of where the receptors are being activated. All right, so there are 6 other hormones that regulate plant growth. So we already talked about auxin, right? We talked about auxin in terms of bending, but it does all sorts of different things. It signals apical dominance, okay, meaning growing from the top of the plant. And so if you've ever tried to do this with your plants, if you want your plants to grow bushier and you cut the top off, your plant will invest more energy in these lateral shoots over here, okay? So if you cut off the top, the tip is not sending auxin down anymore, and then these plants are not, these these lateral shoots are not being, or axillary buds are not being repressed, and they form lateral shoots. Yeah, so auxin signals where the cells should be on the root shoot axis. So when the plant is growing, it tells the plants, you're the root and you're the shoot. And it also is involved in leaf abscission, so we'll come back and talk about that in a bit. Cytokinin is a plant hormone that promotes cell division and extends leaf lifespan. And then gibberellin is a plant that stimulates plant growth. So, for example, this is a plant that has, an allele for dwarfism, and you can actually rescue the dwarf plant by applying gibberellins to it, and it will cause them to grow much faster. And if you put gibberellin on seeds, it will often trigger seed germination. All right, the fourth hormone is ABA, which inhibits plant growth. It also inhibits seed germination, so it stops seeds from germinating, and it also closes stomata cells. Brasinosteroids promote growth and regulate body size of plants. And then ethylene, which you probably have heard of before. This is a hormone that promotes fruit ripening. So if anyone's ever told you, if you have like apples or, some kind of a fruit that's not ripe and you want them to ripen, put them in a bag with a banana. And that's because bananas produce lots of ethylene. Also, like if you have a ripe banana and you don't want your underripe bananas to get ripe, don't put them together with ethylene. Ethylene causes flower fading and also promotes leaf abscission, which means leaves, just unattaching themselves from the tree, basically. All right, so let's talk more about leaf abscission because it involves several of these hormones at the same time. So during the time of the year when you have healthy growing leaves, you have high levels of auxin, kind of at the end of the petiole of the stem of the leaf. And these high levels of auxin are insensitive to ethylene, so you could expose this leaf to ethylene and it won't do anything. Okay? Let's see. Yeah, so the auxin is transported from the leaf to the petiole to the stem. As the leaf becomes older, or as it becomes colder outside, or the days become shorter, less auxin is produced by the leaf, so less auxin is being transmitted down the leaf through the petiole to the stem. And so as auxin levels drop, you get an area here, the abscission zone of the leaf petiole, that becomes more sensitive to ethylene. And so that activates enzymes that weaken the walls of the cells in the abscission zone, and eventually the cell walls degrade and become very weak, and the leaf will fall off. If you were to, so there's the leaf falling off. If you were to interrupt this process by applying cytokinins, you can actually reverse the process and extend the leaf lifespan. Well, obviously not over here, it's too late. But if over here, if you expose this plant to cytokinins, then it would become less sensitive to ethylene and it will not fall off. So cytokinins and ethylene can have opposite effects on leaf senescence. So hopefully in this chapter, you learned about signal processing in plants, a little bit about how plants respond to light, gravity, wind, and touch to cause developmental movements or tropisms, and also developmental changes, and looked at how hormones are involved in youth, maturity, and aging. We didn't really talk about how they defend against pathogens in herbivores, so don't worry about that part.